know about your cell membrane is that we generally refer to it as the fluid mosaic model. And those words aren't chosen just for fun. They're chosen because they describe what the cell membrane looks like or how the cell membrane acts. So the first thing I wanted to do was describe to you why we would use those two words. Oh, where'd my thing go? So first of all, why do we call the cell membrane fluid? The reason that we would call the cell membrane fluid is that the membrane can separate to form vesicles or vacuoles. So I'll just pause so everyone can write that down. So I want you to think of the cell membrane not as one solid structure, but actually like it's a bunch of little pieces, which is really what it is. And that's what the picture down below illustrates. And I'll talk about the components that are in your cell membrane. But for now, you need to know that it does not stay static. Static means that there's no change, there's nothing moving, there's nothing going on. And the cell membrane is constantly changing. Pieces are added to it, and pieces are taken away. The other thing about the cell membrane is that we call it a mosaic. The reason it's called a mosaic is because when you look at it, it's not one type of molecule over and over again. There's a whole bunch, you'll see those little red balls, but there's also a bunch of proteins distributed throughout the membrane. So because of its appearance and what it can do, we call the cell membrane a fluid mosaic. Would anybody like to ask any questions about those two terms? So then what we'll do next is we'll talk about the things that are in this illustration so that you know what each component of the cell membrane is. So we have a few different pieces, and I'll explain each one of them in turn. The first piece is called the phospholipid bilayer. This section here is the phospholipid bilayer. Now, in the phospholipid bilayer, there's a couple of things going on. You have phosphates that face the outside and lipids that face the inside. And it's called a bilayer because there are two layers of it. So when I look at the picture of the cell membrane, and I'll just enlarge a little bit, I see a whole bunch of circles with like two little dangly legs on them. The little circles are the phosphates, the little legs are the lipids. So hydrophilic and hydrophobic have to do with water. And it's the second part of the word that tells us really what's going on. Something that is hydrophilic is water-loving. Something that is hydrophobic is water-hating. And so the reason the phosphates and the lipids are arranged the way they are is because phosphate is hydrophilic and lipids are hydrophobic. Phosphate likes water. It wants to touch the water. And since cells are full of water and cells are surrounded by water, phosphates will always be on the outside surface of the cell membrane and the inside surface of the cell membrane. And the lipids will be hiding in the middle away from water because they don't like it. Does anyone want to ask any questions about the phospholipid bilayer? So the next thing we have in the picture are proteins. First, we have peripheral proteins. Peripheral means that they're on the side. And so I'll show you an example of a peripheral protein. This protein is peripheral. It's embedded on the outside, and you'll notice it doesn't go all the way through. So it's embedded on the outside, and it's used either as an identity marker, so something that identifies like, hey, I'm a cell of this particular type. So my cells would have something that are like, hey, I'm a Mrs. Baxter cell. How are you doing? There's something on the outside of the cell. They can also act as receptors. That means that some molecule, like a hormone or a neurotransmitter, could bind to them and tell the cell what to do. Now, on the peripheral proteins, we also have carbohydrates. So you'll see in a bunch of places there's these little chains. These are carbohydrates. And just so everybody knows, carbohydrates are sometimes referred to just as sugars. And I have the bad habit of sometimes using one word and sometimes using the other, because in my brain they sound the same. But sugars are carbohydrates. That's what type of molecule they are. You'll notice that they're always sort of a hexagon or a pentagon shape in a big long chain. They are usually attached to those peripheral proteins to use or to be part of the identity marker. Now next, you have membrane proteins. A membrane protein would be one like this guy over here. A membrane protein spans the entire width of the membrane. So you can see it from the outside and you can also see it from the inside. And they transport material in and out of the cell. Now they do other things just like the peripheral proteins do. They can receive signals, so they can be receptors. They can help recognize other cells. They can help perform enzyme activity. The thing that we'll focus on, though, is the fact that they transport material in and out of the cell. Now, how would they do that? Well, if you notice on the picture, the protein that I circled has a channel. There's like a space, an opening down the middle of the protein where certain molecules could pass. Now, because of these proteins and because of the nature of the membrane, we call the cell membrane semi-permeable. Semi, like partly, not all. And permeable means that things can get through. So if the cell membrane is semi-permeable, that means that sometimes substances could get through the cell membrane. Now I'll just pause here. All right. Would anybody like to ask any questions about the pieces that are part of the cell membrane? Now I mentioned that the membrane proteins help make the cell membrane semi-permeable, and they're not the only thing. Substances can move directly through the cell membrane, and so I'd like you to notice there's space there. Those molecules are not all one structure. There's space in between them. Small molecules can go right through the cell membrane. So things like water, oxygen, can just move right through the cell membrane in and out of them. The membrane protein is usually for large or charged molecules that have a hard time moving right through the cell membrane. So this is what the cell membrane looks like. These are the pieces that it has. And our goal is to talk about what this has to do with transport. And so what we'll do next is look at the other side of the page that has the beautiful flow chart about all of the types of transport that exist. So I made this little chart to summarize the ways that things can be transported in and out of a cell. So I started out with transport across a membrane that is semi-permeable. Now we already defined what semi-permeable is. I just wanted to repeat it there so that you knew it's a word that you would have to use. The cell membrane is semi-permeable. Now the first categorization that we'll make about transport is whether or not it is active or passive. Active transport uses what we call ATP. ATP is your body's energy molecule. And all living organisms make use of this or something very similar. It's the molecule that provides energy to cells. During active transport, we make use of a membrane protein all the time because in active transport, it is difficult to get things to move a way that they wouldn't naturally move. The other thing that's important to note about active transport is which way the molecules will move. In active transport, molecules move from high, whoops, don't write that. That's just me not thinking straight. Molecules move from low to high concentration. That means that instead of molecules spreading out, they all bunch up in one particular place. Now that's different than passive transport. In passive transport, molecules go from high to low concentration. That means that they spread out. So if there are a bunch of, let's say, oxygen molecules all in one side of the plant cell, because that's where the chloroplast was doing photosynthesis, they will spread out. In passive transport, there is no ATP necessary. It happens naturally. You do not need your cell to use up energy for this to happen. Now you'll notice that I'm not writing something about a membrane protein, and that's because sometimes you use one and sometimes you don't. And so I'm not going to put a general statement there. Now up until that point, does anyone have any questions about the difference between active or passive transport? So, if we go a little further down, we'll start with the active transport types, and there are two of them. And what I'd like to do is together draw one picture that actually represents both types of transport, depending on which way you look at it. So I'm going to imagine that I have a cell. I'm not going to be very artistic with my drawing here because I can't. And I'm going to imagine that up here is a little vacuole with food in it, so I just put an F. And that cell wants the food. It wants to consume whatever particle that is. So here's what will happen. My cell will make a little pocket. So remember, the cell membrane is fluid. It can change. And so all that's happened here is the cell membrane made a little dip. And that little molecule of food is going to go into that little dip that got made in the cell membrane. Finally, the cell will close up the cell membrane around the piece of food. So if you look at this, I think it kind of looks like a Pac-Man eating something. It opens up, the food comes into the cell, and the membrane closes around it. So what's really happening is these two sides of the membrane come up around it and sort of pinch together and reassemble themselves so that you still have a cell membrane, but now that piece of food that the cell wanted is inside of it. So like it ate it. Now he's laughing, but literally this is called cell eating. And so I'm going to give you a couple of words. This picture that I drew goes with endocytosis. And there are actually two subcategories of endocytosis. One of them is called phagocytosis, which is cell eating. And one of them is called pinocytosis, which is cell drinking. Now I'll tell you how I remember this. You guys don't know what alcohol tastes like or that it exists because you're not old enough, obviously. But there was a kind of wine called Pinot, and the cell is drinking, and it's called pinocytosis. That is how I, in my adult brain, who knows about alcohol, remember it. I know you guys don't even know what it is. You've never tasted it. You've probably never even seen it. But this is endocytosis. Now the difference between phagocytosis and pinocytosis is really the size of the molecules. If the molecule is large, we refer to it as phagocytosis. If the molecule is small, we refer to it as pinocytosis. Now, does the molecule have to be food? It does not. This little molecule here could be food, or it could be some sort of invader that the cell is trying to attack. And so if you plan on taking Biology 20, one of the units we discuss is your circulatory system and your blood and your immune system. And you have cells whose job is to do this, but not to food, to attacking bacteria or foreign organisms that are making you sick. Now, what is exocytosis? Exocytosis is this exact same thing, but the other way around. Exocytosis is when your cell has something that it wants to get rid of. If this was exocytosis, I might have some waste, something that my cell digested already that I want to get rid of. And then that waste would get pushed out of the cell the same way food came into the cell. So endocytosis and exocytosis look virtually identical. They're simply in the opposite order. So I'll just pause. On the other side, we have our passive transport. Now here's the key difference in terms of why your cell would use one over the other. Passive transport lets molecules move in their natural direction. Nothing has to happen for passive transport, it just happens. For active transport to happen, your cell has to use energy, so there needs to be a really good reason that your cell wants to take all of a certain particle and put it in one place. So when I think about active transport, I think about nutrients and waste. My cell would want to have all the nutrients inside and all the waste outside. My cell wouldn't want waste to be evenly distributed throughout the cell. The same thing with nutrients. It wouldn't want all the nutrients to be outside of it. Now, passive transport happens naturally. That means your cell is not in control of it. So sometimes passive transport can be very helpful, but sometimes it could cause harm to a cell because it's happening naturally. We have three kinds that we'll talk about. The one we'll focus on is diffusion. Diffusion happens across the membrane. So I'm not using a protein here. Things are moving in and out through the spaces in the cell membrane all on their own. Facilitated diffusion is the same process, but it uses a protein. It's for molecules that are too big to go across the cell membrane by themselves. Osmosis is very similar, but osmosis is specifically for water. And so here's something that I'd like to note that's different between diffusion and osmosis. In diffusion, we are talking about particles. And the particles move from a spot where they have high concentration to a spot where they have low concentration. In osmosis, the same thing is happening, but it's happening with water, and so it seems like actually the opposite thing is happening. Water always moves towards high particle concentration. Now that sounds like the opposite of passive transport, but if there are lots of particles, that means that there's not very much water. So water is still moving from a place where it has a lot of concentration to a place where water, has a low concentration. But if we're looking at particles, which we usually are, water tends to move the opposite way. Water always moves to the place where there is higher particle concentration, trying to dilute it. Would anybody like to ask any questions about the descriptions of diffusion or osmosis or facilitated diffusion? So then down at the bottom, we have a few words that have to do with concentration gradients. Now, a concentration gradient is a difference in concentration between two places. And usually that is inside and outside of the cell. So typically we are comparing the concentration inside of a cell to the concentration outside of a cell, trying to figure out whether particles will move into the cell or particles will move out of the cell. And so when we're talking about a concentration gradient, there are three different ways that we could describe it. If I say that I have a hypertonic solution, that's usually the word that we would use, a solution, that means that its concentration is higher. And it has to be higher than something that we're comparing it to. So you can't just say the solution or the cell is hypertonic. You have to say hypertonic compared to something else. Now, if we use the word isotonic, we are saying that concentration is equal to the concentration in some other location. Now remember, we're always comparing two places, so you can't use one of these words without telling me the two places that you are comparing. Hypotonic means that concentration is lower than some other place. So I'm just going to pause here and make sure everyone has a chance to write these. Now we're going to pay particular attention to osmosis as an example of diffusion, because for a living cell, how much water is available is usually crucial to its survival. And so on this page, you have a series of pictures, and I'll just show you how they're categorized. The top row are all pictures of animal cells, and the bottom row are all pictures of plant cells. The first column is what happens when you put the cell in a hypotonic solution. The middle row is when you have an isotonic solution. And the far column here is when you have a hypertonic solution. And so we're going to focus on why water would move in or move out of a cell. Now, in the first box, where we have an animal cell in a hypotonic solution, what we're saying is that this has a low concentration and this has a high concentration. Now, when we say concentration, we mean of a particle of some type. So just for saying's sake, let's use salt. Salt is a particle that's often found in living organisms. So we will talk about salt. Now, when I put a cell, an animal cell, into a solution that is hypotonic, meaning there are less particles outside the cell than there are inside the cell, here are the things that would want to happen. The salt, if it could, would move out. But sometimes particles are too big or they can't move through the cell membrane, and so very frequently, instead of the particle moving, water moves. So water always moves towards high concentration. Water is trying to even the playing field, dilute the higher concentration. So if you think about this in terms of a substance that you know about, let's think about making Kool-Aid. Kool-Aid is delicious, and you know that if you want really strong Kool-Aid, you either need more powder or less water. And so we're focusing on those two ideas. If I want these two concentrations to become equal, I either need less salt inside the cell or more water inside the cell to even things out. Now, when water moves in, what is the result? Eventually, lysis. That means that the cell will burst. It will get so full of water that it will burst and then it will die. So this causes a cell to die. Now that doesn't mean that 2 seconds in a hypotonic solution means death for an animal cell. But it does mean that if it's in a hypotonic solution too long, this could happen. So I was watching, I think it was the show called 1,000 Ways to Die, and there was an episode of it about a radio who was getting people to do this contest where they drank as much water as they could without peeing, and people were talking like, oh, water can't kill you, water's good for you. Water could kill you. If you have too much water, you will drown out your cells because your cells are constantly trying to maintain this equilibrium. If you drink a bunch of water, you're making a hypotonic solution, and eventually your cells will get so full of water that they would burst. So you might remember in grade 9, I don't know, who knows what you remember, you don't, but I know that in Unit C in environmental chemistry, there's this thing called LD50. Do you guys remember hearing about this? Now, LD50, or lethal dose 50, is the amount of something that would kill half of the people that took it. Water has an LD50, it's just really, really high, because it would take a lot of water to kill you. Now, what happens to a plant cell in a hypotonic solution is not the same thing. I'm going to write the word good here so that you know that plants like this. This doesn't kill them. Now, when you have a plant cell that is in a hypotonic solution, think about spraying vegetables at the grocery store. In the grocery store where there are fresh vegetables, there are little water misters over top of them that are constantly spraying them with water, and this is why they're doing it. When a plant cell is in a hypotonic solution, same thing happens. Water goes in, but plants have a cell wall and this large central vacuole. Plants have a ton of place to store water, and they want to. When a plant cell gets really full of water, we call it turgid. And this is what makes vegetables crunchy. So if you have, like I found in my fridge yesterday, some limp celery where you pick it up and it just bends over, it's totally not crunchy and I wouldn't want to eat it. The reason my celery got limp is because there was not enough water in all of the cells to keep them puffed up so that it feels crunchy when you bite into them. So this is good. Plant cells like this. Now the middle column is an isotonic solution. In an isotonic solution, notice that we're showing water coming in and water coming out. That is because the concentration would be equal. There would be the same concentration in and out of the cell, and so water would move back and forth freely. Actually, in all of these examples, water would move in and out. It's always doing that. It's just that in the first example, way more water moves in than out. And so we just talk about the water moving in. No, I want you to focus on the water going in. The idea is that water is constantly going across the cell membrane all the time, but then in hypotonic solution, it's primarily going in. Now, the words that we would use to describe cells in an isotonic solution, for an animal cell, there's not really a word. We would just call it normal. For a plant cell, there's a word. We call it flaccid. So when I described my celery and how it was kind of limp, it was flaccid. It was not turgid anymore. So notice the difference in the picture. The turgid picture looks all puffed up, and the flaccid cell just looks regular and rectangular. Now in a hypertonic solution, and I'm going to give you an example, let's say that now we are in ocean water. That's our solution. Ocean water has a bunch of salt in it, generally a much higher concentration of salt than normal living cells of terrestrial organisms have. In this case, water is going to leave the cell because the concentration is higher outside of the cell than inside the cell. And so the result of this is bad. So for both plant and animal cells, this will mean eventual death. For an animal cell, the way we would describe what is happening is with the word shrivel. The cell will literally shrivel up and die. It will lose all of its water eventually, and it will no longer be functional. For a plant cell, we refer to this as plasmolysis. Now, if you notice what's happening here, it looks like the cell wall and the contents of the cell are tearing away or ripping away from each other. The cell wall is actually quite rigid, and so unlike the cell membrane, it can't just shrivel up and go away. The cell wall is a pretty solid structure, but the contents of the cell are trying to shrivel up, and so it looks like they're ripping or tearing away from the cell wall. The word lysis means ripping or tearing, and the word plasma is always talking to the cytoplasm or the fluid that's inside the cell. Annalise. No, for the plant cell. And shriveling is for the animal cell. So, if I ever ask you, Should you drink ocean water if you're shipwrecked? You would say no, you will die faster drinking salt water than if you drank no water at all. You would be better off to drink your own urine because at least that has actual water in it. Does anyone have any questions that they'd like to ask here?